we're doing the citric acid cycle, and remember there's a step 0. You can't go directly from glycolysis into the citric acid cycle. So pyruvate does not directly go into the citric acid cycle. It has to be first transformed into acetyl-CoA, and that was done by the Pyruvate Dehydrogenase Complex, and it really is complex and it's huge. I've got a cryo-electron micrograph of it in just a moment, but it requires 5 different factors along with it, okay? And we'll find out that since the dehydrogenase, it does make NADH, and as I mentioned in the past, we'll find out in chapter 20 that each NADH will go on to make 2.5 ATPs, okay? All right. And so this is a better representation for it. So we have, first of all, what is CoAsh? This is coenzyme A. It's acetyl-CoA whenever, well, later on when it becomes acetylated. Okay, so it requires acetyl-CoA. It requires NAD plus NADH, TPP, lipoate, and FAD. Okay, and it is this huge complex. The products for each of these enzymes actually does not leave the complex. They just get shelled from one part of the enzyme complex to another part of the enzyme complex. And ultimately, it is a decarboxylation reaction, and you get acetyl-CoA in the end. When we last left off, we were talking, I do believe, we started to go through what all of these different things were. And we actually saw TPP, thiamine pyrophosphate, in the past. And so does anyone remember what does TPP do? What's it important for? What kind of things? Because you should know what your cofactors do, just like NAD plus, NADH, and FADH2 are for redox reactions. What does TPP do? It helps transfer something. Not electrons, that's a redox reaction. Pardon? No, that was a good guess though. It's for acyl, acetyl or acyl compound. So of course, if it's only 2 carbons long, it's considered an acetyl group. But it also, we're going to see it later on with like fatty acid synthesis and things like that, because it's great at transferring acyl groups. Okay. And the way it does that is it activates it, and we've got a picture coming up in just a moment of a little cartoon depicting all of this. Now, some of the other ones that we haven't encountered or haven't encountered as often include coenzyme A, and then they always abbreviate it as CoAsh, and then if it becomes acetylated, it's acetyl-CoA. The reactive group is just a thiol whenever it's coenzyme A. That's why they say CoAsh. This actually comes courtesy of a cysteine, eventually, or originally, I should say, a pantothenic acid, and then, of course, this is the adenine group. And for those in neurochemistry, we actually talked a little bit about this because of the fact that it was Lorraine's topic on Monday that discussed coenzyme A synthesis. Okay, and coenzyme A is also really good at transferring acyl groups, and so we're going to see it with respect to fatty acids and either beta oxidation or fatty acid synthesis. This right here is your lipoic acid lipoate. Okay, and it's, once again, it does do redox reactions. However, it does it via acetyl transfers again. And so what happens with it is it's a covalently attached. It's not like a ionic interaction or something like that with respect to coenzyme A. It gets covalently added onto the enzyme via a lysine group. Then this is lipoic acid, and when it's oxidized, it's a disulfide. When it's reduced, if it's fully reduced, then it's 2 free thiols. And that's the reason why you can use it as a great nucleophile for the sulfur to go and smack the carbonyl and transfer it from one place to another place. Okay. So this is the cartoon of how it works. So like I said, it just kind of gets shuttled along this big complex, and to me it kind of looks like, in a moment you'll see why, I think it looks like honeycomb cereal. But the TPP acts first, and so this one is a dehydrogenase reaction. So what happens is it decarboxylates it, and then the two carbons, which will become the acetyl group, gets added to the actual TPP part. Then it just goes and it gets shuttled to the lipoic acid. Lipoic acid then, what it does is this is where the actual coenzyme A gets added on. Okay, so that's why it's important. And then the acetyl-CoA exits the enzyme complex. But this would short circuit if we didn't have a way in order to regenerate the lipoic acid and the TPP and everything else. So what happens here is that the lipoic acid then gets oxidized via the reduction of FAD. So that's the reason why it does require an FAD. And then so now it can go back in order to hook up with the next TPP that's already has a hydroxyethyl group attached to it, and it can repeat this process. But we have to then get our FAD back, and so the way that the FAD comes back is the fact that this is the actual dehydrogenation reaction, where the NAD plus comes in and then NADH leaves, and so that's how you regenerate your FAD. So they just kind of pass it off from player to player, except you have to realize that the acetyl-CoA itself never comes into contact with the NADH.
So all of these cofactors never actually leave the complex.
None of these, I should say, the coenzyme A, because that's technically not a cofactor. In this instance, it's actually one of the reactants. But none of the intermediates leave. So, like the product for the first enzyme, you don't have to worry, I did it wrong. You don't have to worry about the hydroxyethylated TPP leaving, because what it does is it automatically then just goes on and gets transferred over to the lipoic acid. And you don't have to worry about the lipoyl lysine leaving either. Well, that one's covalently attached, so you don't have to worry about it leaving anyway, but then the coenzyme A comes in and then it takes off the acetyl group and then you regenerate the rest of it. But yeah, it's the product of the prior reaction does not leave before it goes into the next step. So this is what it looks like. Like I said, it's huge. It's a multimeric protein. And you can actually see it through microscopy. So that's why, to me, it kind of looks like honeycomb cereal. But yes, it is very, very, very large. 50 nanometers across is a large protein complex. So, all right, now we're going to actually start into the citric acid cycle. So we've made the acetyl-CoA from the pyruvate via the Pyruvate Dehydrogenase Complex, and so technically that's what I would call step 0, because it's not a part of the circle, but it has to occur in order for the circle to occur, to go on. So this very first step is actually the production of citrate. Okay, and so if you notice, we have oxaloacetate that comes in. Okay, where have we seen oxaloacetate before? Part of the malate shuttle, which is a part of what metabolic pathway? Gluconeogenesis. And we've also, I have talked about, we actually haven't seen this metabolic pathway, but where else can we get oxaloacetate? I mentioned it. It's similar to the ways that you can get alanine and pyruvate. It's aspartate or aspartic acid. Okay, this is the keto acid form of, if it's amino acid, it would be aspartate. And so here we have OAA, or oxaloacetate, hooking up with the acetyl-CoA in water to make citrate, and this is where we get this tricarboxylic acid idea, because there's 1, 2, 3, 1, 2, 3 carboxylates there, and you get your coenzyme A back to go on and to do this again. So what class or classes of enzymes could this possibly be? It could be an acetyltransferase. What else could it possibly be? What else are we doing besides just transferring the acetyl group? We know it's not a cyclase or nuclease or protease. Is it a dehydrogenase? Do we have the stuff there for it to be a dehydrogenase? No. Is it a kinase? No. So what else could it possibly be besides solely being an acetyltransferase? What are we doing here? We start with oxaloacetate and Acetyl-CoA, and we make citrate. Synthase. And so this is citrate synthase. We are building, and by the way, please note that this is one of the checkpoints. It's irreversible. In a way, pyruvate dehydrogenase complex itself is also a checkpoint because it's allosterically controlled, but it's not part of the circle. It's a way to control it from coming into the circle. But step one, citrate synthase, is the checkpoint. Okay, so it's allosteric. What do you suppose are going to be some of the things that would make sense that it would be inhibited by? Because two of these I could argue that it makes sense that why they would inhibit this enzyme for the citric acid cycle. You could make the argument for citrate. What else would it make sense? Just think in the big scheme of things. Pardon? ATP, and ATP is one of them. What else is a big scheme of things that would inhibit it? If you start to have a lot of this, it means you've got plenty of energy. That's why if you have a lot of ATP, you've got plenty of energy. And it's not alanine. Pardon? NADH. Those are the two that makes the most sense. ATP and NADH, because remember the whole purpose, or one of the major purposes, I should say, for both glycolysis and the citric acid cycle is, you're making these reducing equivalents of either FADH2 or NADH to go on to the electron transport chain. The third one, you guys wouldn't know about yet, and it's succinyl-CoA. Okay, and the reason why is succinyl-CoA is a metabolite that's further down the citric acid cycle. It is a circle. Okay, so that's the reason why it makes sense once we get to it, but right now it wouldn't make sense. So ATP, NADH, succinyl-CoA, those help turn off citric acid synthase. So this just shows you how this is a dimeric protein, so you can see, one of the reasons why I really like this is you can see clearly how it's like the binding site, how they can come on in here and activate each other and react. But this is really cool. This is step 2. And by the way, they're not including step 0, there are 8 steps. This is a dehydration, rehydration. You're going to form isocitrate. Okay, and this one does have so many books show this. This is a 2-step process, and it does have an intermediate that you can sometimes isolate, but it doesn't leave. That's why it's technically all one step. One metabolic step, but this enzyme does 2 things. It does dehydration and it rehydrates. And please note, it uses iron. And it's one of my favorite ones to talk about because of the fact that they call it a Ferris wheel. And so, which, especially those in neurochemistry, we've talked a lot about iron. This is one of your iron enzymes, and the iron plays a very important part here because it's what is utilized in order for it to work as an iron-sulfur center. Okay, and it goes from what's unusual is if you think back to organic chemistry, we'd always talk about how if you start with achiral reagents, you're always going to end up with an achiral product unless you somehow force it to be in a chiral environment. This is the way that we get around that in biology. in vivo because citrate is achiral. And the reason why, I mean, it's pretty simple to see. There, you can cut it right here with your guillotine, and the top half and the bottom half are identical. So that carbon is achiral. However, at the end, when you have isocitrate, which is an isomer of it, it's actually chiral now. In fact, it's got 2 chiral centers. Okay. This enzyme is the one that usually gives people fits. Okay, this is the one that sometimes, because it doesn't make sense. It's named after the intermediate form. It's not named after citrate. It's not named after isocitrate. It's named after aconitate, which is interesting, and I don't know why. There's got to be a good story there, but I don't know it. It's called Aconitase. Remember, we've had aldolase and enolase. And those made sense once you looked at the fact that, you know, they made an aldol and it made an enol. This one doesn't make aconitate, so it doesn't lead the enzyme, but it's named after the intermediate. And so that's why sometimes they show it as steps 2A and 2B on some figures, but it's still the same enzyme that does this. Okay?
Is that also a checkpoint?
No, this is just no. This step 1 is the only one that we've seen so far. All right, and so this has an iron-sulfur center, which, as I mentioned in some of the other classes before, iron can chelate or hold on to their special bonds between metals, like especially transition metals, and their ligands. And so iron really likes sulfurs, especially like cysteines. It also can bind to like nitrogens too. And so, but because of this, it affects its electrochemistry, and so that's why it helps with catalysis many times whenever you're shuttling electrons from one place to another. Okay? And so that's why I wanted to point out that this, it must have gone to sleep or something, because now it's not working, is being held in place by the cysteines. Okay? So it helps with both binding and in the actual catalysis. Okay. This is step 3. This is the second official checkpoint of the metabolic pathway, not including Pyruvate Dehydrogenase Complex. I always want to point that out. So we have isocitrate, and we are making alpha-ketoglutarate, which is sometimes they abbreviate that as alpha Kg. It does have an intermediate, but this intermediate is transient. It doesn't. You can't isolate it from the enzyme. Once it gets started, it's going. Okay. What kind of reactions are occurring here? Pardon? So it does remove a carbon dioxide. So what kind of activity is that? What kind of enzymes do that? A carboxylase or a decarboxylase, right? What else is going on here? Okay, and so what class or classes of enzymes could that be? Dehydrogenase. And so we're going from isocitrate to alpha-ketoglutarate. So what do you suppose that this could be called? What enzyme name could this possibly be? And you are correct. It's isocitric dehydrogenase. So once again, as long as you can learn the logic behind the enzyme names, it helps a lot. It doesn't help you with Aconitase, but it does with isocitric dehydrogenase. Okay. It is also an allosteric enzyme. So what do you suppose would make sense that it would inhibit this enzyme? What are some 2 big players that you would think would make sense that it could be inhibited by? What's one thing? ATP, and ATP is one of them. What's the other one? NADH, and NADH is the other one. And so What would be some of the things that could activate it? This one actually can be activated. And it makes sense for reasons why here as well. What are some of the things that are indicators or good indicators that you need energy? ADP. ADP activates this one. And what else? It's not AMP. That would be a great guess. NAD plus. Okay. Moving right along. Oh, it's lost its internet connection once again. Why is the internet so slow today? There we go. All right. So this is step 4. Step 4 should look really, really familiar. All right. And so we have alpha-ketoglutarate. And by the way, I want to point something out about alpha-ketoglutarate in just a moment. It requires an NAD becoming an NADH. It requires a coenzyme A. Now you're going to get Acetyl-CoA. And it has, well, magnesium, which we've seen before. But TPP, lipoic acid, and FAD. And you remove a carboxylate. So you get a decarboxylation reaction here. So what class or classes of enzymes could this be? A dehydrogenase. Where have we seen this before? Almost the exact same layout. Pyruvate Dehydrogenase Complex. So using logic here, what do you suppose this one's going to be called? This is alpha-ketoglutarate dehydrogenase complex. Okay. And by the way, even though it shows that it's reversible, this is also a checkpoint. Okay. So within the circle, it's steps 1, 3, and 4, and then we also have the gateway of step 0 of being a checkpoint in a way. Okay. And this is the succinate-CoA, which can also, if you start having lots of energy, so your energy is building up, succinate-CoA's concentration will increase, and it's what feeds back, or can help feed back, to turn off the citrate synthase. Whoops. I forgot to ever click on it, so you should see the name, alpha-ketoglutarate dehydrogenase complex. Before we go any further with this, let me see if I can write. Here we go. If we look here, let's see, what does that mean? And let me get a different color. I don't know what color this will be in there. But if we see alpha-ketoglutarate, so it's got a CO2 minus a CH, carbonyl, CO2 minus. Did that all come up there? Okay, awesome. What does that look an awful lot like? Because we're going to find out this is one of the steps that you can feed in, just like we had the glucogenic, amino acids and compounds. There are those that can feed into the citric acid cycle. And one of them is they can feed into this step right here to make alpha-ketoglutarate. What does it look an awful lot like? Think back to some of the other examples that we've had, like pyruvate, oxaloacetate. This is the keto acid form of glutamate or glutamic acid. Because what happens is if you just take that ketone and you convert it to where we have the carboxylic acid, a CH2 group, a CH2 group, and that's your C alpha is now an NH2 or NH3 plus. CO2. See, that's this glutamate. So that's why it's called the amino acid form, and alpha-ketoglutarate is the keto acid form. And so what class of enzymes can do that? Add the amino groups. So you just go through your enzymes. We have the synthases, the phosphatases, phosphorylases, kinases, transferases, cyclases, dehydrogenases, reductases, oxidases. If someone said it, I got underneath their breath. Transferase, and what kind of transferase in particular is this? Aminotransferase, and what specific aminotransferase is this one? We're going to find out, actually. In fact, it's really, really cool because, well, I don't want to give it away. But I was going to say, you're going to see this, you're going to see alpha-ketoglutarate a lot. It's a major player in your body for metabolites, similar to the way that we've already seen alanine and pyruvate a lot in various things, and oxaloacetate. Okay. Then we have step 5, which is succinyl-CoA, going to form succinate. And please note, this is the very first step of the citric acid cycle that directly makes any energy. Now, we had steps that made NADH, and then NADH can go on into the mitochondria, if it's not already there, and make ATP eventually. But this is the first one that makes it directly. So I have succinyl-CoA making succinate. There's GDP that becomes GTP. Okay. So what class or classes of enzymes could this possibly be? And even though it's showing it being irreversible, this is technically not a control checkpoint for the cycle, by the way. What class or classes of enzymes? What are some of the classes of enzymes that utilize GDP, GTP, or ADP, ATP? Kinases? Are there any others? Phosphorylases wouldn't. Is it synthase? What? Synthetase. Okay, and so what could this one be? Well, this is a synthetase. So that one I would give you almost all the points. This is actually named for the reverse reaction. Succinyl, because and if you think about it, and you give it, it kind of makes sense in the sense that it does require the energy of GTP for it to go backwards to become GDP in order to make the succinyl-CoA. So this is succinyl-CoA synthetase. Okay. So succinyl-CoA synthetase. It's named for its reverse reaction. And then you actually get the ATP using our good friend nucleoside diphosphate kinase, which we've seen before. Remember, that's how we got our UTP back during glycogen metabolism. So, because that enzyme, it takes any of the NDPs and can react it with ATP, and they just swap phosphates. So it goes both directions. But yeah, this is the very first one that you get an GTP/ATP from. Right. Then we have step 6, and I put step 6 and 7 on the same. Let's just review real quick, where does the citric acid cycle occur? Not in the matrix. And I remember I said there's this one step that technically is anchored in the inner membrane. It's step 6. Step 6 is actually the same enzymes in 2 different pathways. Okay, and so it's another way that they interact. So we're going to see step 6 again, and for whatever reason, I mean, you can call it either name. In one pathway, they specific because it's part of a large complex. One pathway they call it what we're going to call it here in citric acid cycle. The other pathway they call it complex 2. I mean, which is really simple. Okay, but yeah, for the citric acid cycle portion of it, it oxidizes succinate to make fumarate, and this happens in the inner membrane. And if you notice, it's essentially at equilibrium. The delta G is equal to 0. Okay, but it generates an FADH2 from FAD to FADH2. So what class or classes of enzymes could this be? Dehydrogenase, and what is it dehydrogenating? Succinate. So this is succinate dehydrogenase. And once again, in chapter 20, we'll find out that this FADH2 will only make 1.5 ATPs, whereas an NADH goes on to make 2.5 ATPs. And it'll make perfectly logical sense in chapter 20 why that happens and why it's important. Now, step 7 is a hydration reaction to get L-malate, which we've seen malate. Where did we see malate before? I mean, it's malate shuttle. It helps out with gluconeogenesis and reducing equivalents from one side of the mitochondria all the way into the cytosol, since NADH cannot cross the membranes itself directly. So this is another way that they all start to intertwine. Right? This one has a special name. So we already had Aconitase. This is the other one that's got the special name. So that's why. What do you suppose it's going to be called? That would be a good guess. But it's not correct. Instead of maltase, it would be... No, that's the sugar. Fumarase. So this one's fumarase. So, yeah. Okay. And last but not least, we have step 8 to get our oxaloacetate. Now, oxaloacetate is what's going to hook up with the next Acetyl-CoA in order to make citric citrate. Okay. Notice we have an NAD plus forming an NADH. That NADH can then go on to make 2.5 ATPs later on. Okay. So what class or classes of enzymes could this be? Dehydrogenase. And if you're going to name it, what would you name it? And this is malate or malate dehydrogenase. And so if you include the step 0, because some books include step 0 whenever they add up all the totals, some books don't. You get directly from the citric acid cycle, you only get 1 GTP, which can go on to make 1 ATP. You get 1 FADH2, you get 4 NADHs, and 3 carbon dioxides for respiration. And but now, if you don't include step 0, then it would only have 3 NADHs. But that's why I put the little asterisk there, just in case you see a discrepancy between books, or even within chapters of the same book sometimes, or something. That's the reason why. But yes, that's malate dehydrogenase. Okay. Awesome. So there's the reaction, like just this is from the old book, but where it just lists it as words, so you can see once again, and then and you can put this on your own, or when I put this on MyFire. Okay, so we have the control points. I've already told you that there's 3 points within the cycle. One was citrate synthase, because it's inhibited by ATP and NADH, which that should make sense. And then the Acetyl-CoA, which is coming from step 4. Okay, as well as citrate. We have isocitric dehydrogenase, which is step 3, which is activated by ADP and NAD+, which are indicators that you're low energy, and it's going to be inhibited by ATP and NADH. And then we have alpha-ketoglutarate dehydrogenase complex, which is a mouthful to say, and it's inhibited by ATP, NADH, and succinyl-CoA, once again, its own product, and it's activated by ADP and NAD+. And then likewise, there's that one that's just outside, like the gatekeeper. And it's pyruvate dehydrogenase, which is inhibited by ATP and NADH. And it's also inhibited by acetyl-CoA. By its own product. Okay. So technically step 0, steps 1, 3, and 4.
That's pyruvate dehydrogenase complex.
Yeah, the complex, yeah, sorry. So there, if you'd rather like to see it as a little cartoon, there it is. Try to appease all learners. This is the important idea here, and so that's why a lot of times I may ask questions and I'm looking for your logic, and so then you want to explain it, but you also don't need to write like 3 pages on a single problem either. But if you're in a resting metabolic state, if your cell is, or that cell is, then it's going to use very little energy. So you're going to have a lot of ATP and a lot of NADH, okay? So that makes that ratio of ATP to ADP, or NADH to NAD plus, too high. And whenever that happens, that's why you want to regulate and turn down the citric acid cycle as you do glycolysis. And you're going to turn on gluconeogenesis. If you're actively in an active state, then you're going to need more energy, and so the ratios then get reversed. Okay? And so that's why a lot of times you just have to try to think about, okay, how is this affecting the overall ratio of ATP to ADP, or NADH to NAD plus. All right, and so we're seeing how, and I've been trying to point these out as we go along. So in particular, the ones that at this point you really should know: alpha-ketoglutarate and oxaloacetate. Whoops, and it's not on here, but alanine, which is the one that goes right there to make pyruvate directly. Those are obvious because those are the aminotransferases from the keto acid to the amino acid form. We're going to find out how fatty acids make acetyl-CoA. It's in the future chapter. And then proteins are broken down by proteases to give off their individual amino acids. And we'll talk a little bit about the succinyl-CoA, fumarate, and malate later on in future chapters, and how they can all feed into the citric acid cycle in and of itself. Okay, I'll show this last figure. This is just a cartoon showing it, and the TA that's up here, this TA, is for the old form, the name transaminase, which sounds really cool, right? Like a hot rod. But it's aminotransferase. But I really like this because, what? I know, transaminase. Isn't that awesome? So I really like this because it's trying to show you how things get into the mitochondria where the citric acid cycle occurs, and versus in the cytosol. And so we have already talked about, for example, aspartate here with aminotransferase making oxaloacetate, and that was malate. That's malate dehydrogenase. Okay, then the malate can go on through, and then it can hook up with the citric acid cycle to go on with malate dehydrogenase on the inside to oxaloacetate and so on and so forth. That's just one example, and we had the example already with the alpha-ketoglutarate, and then we're going to find out about the succinyl-CoA, and then we've already talked about pyruvate coming either from glycolysis, from alanine, and then there are more indirect ways. Okay, so that's one reason why I like this little picture of that, and they do much better job at drawing the mitochondria than I do. So, okay.